I'd like to start the day off by introducing somebody who is a really cherished colleague here at UCLA, who's a professor, associate professor in the Division of Human Genetics and Medical Genetics in the Department of Pediatrics and in the Department of Psychiatry. He has many roles, and really one of his main roles has been to serve as a partner with us in terms of the clinical genetic approach to children with autism. And it's kind of remarkable what he and Dr. Geste have done together, and his expertise coming forward in beginning to kind of pull autism into the kind of mainstream of pediatric genetics. He's a really esteemed clinician, colleague, as well as researcher. So without further ado, I'd like to introduce Dr. Julian Martinez, who's both a MD and a PhD, which reflects both his clinical and research hats. And this is the first time we've actually had the pleasure of having Dr. Martinez here. So again, thank you, Julian.
Well, thank you for the kind introduction and to the organizers for inviting me to participate. Just to begin, I have no disclosures. The intent of the talk is to, first of all, introduce, although I hope that it's not a surprise to most of the people in the audience, a little bit about what autism spectrum disorders are and how we approach clinical care by incorporating genetic evaluation and diagnosis to really provide answers to families that have for a long time really tried to seek either interventions or an explanation for why their children are having the neurodevelopmental challenges that they have. So ASDs, as you all know, are a group of neurodevelopmental conditions in which there are impairments in social interaction and communication. There are restricted, repetitive behaviors that can go along with that presentation. The incidence has increased significantly over time, as high as 1 in 68, with a sex bias towards males. And we know from studies that Dr. Geschwin and many other laboratories have pioneered over the years that there is a genetic contribution that has emerged over the last decade. And in addition, an environmental contribution that a lot of experts have also identified in both animal models and in human epidemiological studies that make sort of combine to give the phenotype of autism as we understand it. So when we talk about the genetic basis of ASD, we know that it's very complex. There are more than 800 genes that up to date have been carefully curated that when altered, may present with autism spectrum disorder findings. And each gene probably accounts for a small proportion of each of the cases that we see. But when we talk about patients with ASD, we also have to take into consideration that developmental delay is also a usual co-diagnosis many times. And we broadly define it as an age-matched deficit in 2 or more categories of developmental milestones, whether they are language, motor, social, or other activities of daily living. It's usually diagnosed before the age of 5. And again, there is an overlapping literature that demonstrates that the genetic etiology for both developmental delay and autism is kind of it's merging, it's overlapping. So when we look at patients that present with autism, we want to be very careful because we want to know to what extent is intellectual disability or developmental delay kind of a cohabitant of the phenotype. And when we look for clues for what might be likely a genetic etiology, knowing that can be very useful. So what do we know about the genetics of autism? So we know from very early studies, and some that have updated the numbers recently, that when you look at twins, particularly identical twins, that there is a higher concordance when they're monozygotic than when they're dizygotic, meaning that if one is affected, more likely the monozygotic identical twin will also present with ASD symptoms. So that gave us sort of an initial clue that there is some contribution from genes. But when we looked at patients in the clinic, we found, and us and many other groups have identified that when patients present, you can almost separate them into 2 broad categories. A child that presents with primarily the social and communication deficits, or sort of isolated autism symptoms, and then the child that presents with developmental delay and sort of more other comorbidities. And it's actually the one where we have this syndromic type of autism where you have the likelihood that this might be representing part of a rare genetic disorder in which autism and other comorbid comorbid symptoms are part of the presentation. So that has led to us to believe that autism is not really one condition, but actually many under one umbrella, and that many patients will present with many similar symptoms, and it's very difficult to determine which genetic etiology they each have because their clinical presentation is so similar. But when you really dig in, you actually find out that many genes that are talking to each other are presenting when altered in many similar clinical ways. So what we also know is that the symptomatology of autism includes many other comorbid symptoms, whether it's seizures, motor concerns, hypotonia, the presence of accelerated brain growth or poor brain growth, and all of those clues help us understand which type of syndromic etiologies for autism we might be dealing with. One of the other challenges of evaluation of patients that have come with a neurodevelopmental diagnosis is that we know from very large studies of rare genetic diseases that they come with a lot of history and baggage that is really weighing on them. We know that 25% of patients wait up to 30 years sometimes to actually get a final diagnosis that brings brings an explanation to the family. Many of them, up to half of them, receive an erroneous diagnosis initially that then later on gets corrected. Many of them have to travel very far distances to get the care and the evaluations that they need. And only half of them, if not a third or a quarter, receive some proper genetic counseling for proper family planning and familial testing. So this is part of what we call the diagnostic journey and the odyssey that families sometimes go through, where they're provided a clinical diagnosis, but the real genetic etiology is not identified many years later because they're not encouraged to pursue that workup. And we know that when you look at just admitted children in the hospital, up to 75% of cases that we see admitted in the hospital units will have some genetic contribution to their clinical presentation. So we have a high incidence of comorbidities that require hospitalization, and therefore genetic evaluation in which these patients may actually require more care and more specified and targeted personalized care because of their uniqueness in clinical symptoms. So when we evaluate someone in the genetic clinic, what we're trying to do is achieve or verify that the clinical diagnosis of autism is actually accurate, provide a geneticist or a genetic counselor to provide both pre-test and post-test counseling, and then offer genetic testing itself that would lead us to the final etiology. So the clinical diagnosis requires us to work closely with individuals at either regional centers or in the community, fellow neurologists or psychologists who constantly provide, or developmental pediatricians who provide diagnosis of the autism spectrum entity itself. Then when the patient is referred to us, we perform a physical exam and we try to put physical finding with the family history and other clinical criteria to identify what are the patterns or phenotypes, which is a collection of clinical symptoms and physical findings that the patient has. Do they present with a specific pattern that we can recognize? And as I just explained earlier, the intent initially is to figure out, are they presenting with isolated autism or are they presenting with autism that's presenting with other symptoms, whether it's physical or additional clinical ones. And the intent of doing the genetic testing is to guide diagnosis, is to provide medical surveillance because many genetic conditions will have associated problems that may develop down the road that we can implement a surveillance program for. Provide family counseling for family planning purposes and to identify other potentially affected or at-risk family members. Provide advocacy and support and resources. Prognosis, what does this mean down the road? What are the other things that we need to check for? Identify other potential comorbidities, and then personalize, ideally, the treatment based on the genetic cause that's identified. So the goal is to better understand the clinical course of genetic conditions, reach out to community physicians and families to make them aware of the availability of this workup, provide counseling to address uncertainty of what caused the symptoms and whether it will happen again in future pregnancies. Many times more than one affected child is born and in affected families before they actually reach a diagnosis where they can actually implement and use for future family planning, anticipate complications, and develop a treatment plan that's unique to the individual or the family. So that requires careful family history, prenatal history, exposures during the pregnancy, developmental history of the patient, physical exam, and then proceeding with genetic diagnosis that's either targeted or more broad based on the clinical findings. So what are the things that we look for when we're looking at the history and even the physical exam in patients? We're looking for one or more major abnormalities. So for example, a child that comes in with having been born with a heart defect, that would be a major birth defect, or 3 or more minor malformations. So we call those dysmorphic features. So those are little things that when we look at someone, they look a little bit different than most people in the population. And when you see 3 or more of those minor malformations, the incidence or the likelihood that there will be something major, whether it's a brain abnormality or some other congenital defect internally, goes up as high as 98%. So using external physical cues is very telling to predicting the possibility or the likelihood that we'll have a genetic diagnosis. In addition to, are they growing well? Do they have problems with seizures, skin, bones, genitalia, and immune or behavioral problems? So what do we know about the genetic basis of autism? We know that up to 20% of cases will have what we call a chromosomal abnormality, meaning chromosomes are the packets where genes are housed. And what can happen is that neighborhoods of those genes can either go missing or be copied or present in more copies than there should be. So how do we assess a patient clinically for the presence of those chromosomal abnormalities? The technology has changed dramatically. When I trained, what we used to do was send a karyotype, which is just taking blood and looking at the chromosomes under a microscope to see if there were any egregious large pieces of chromosomes that were missing or extra. Now we have a more sophisticated approach that uses a whole genome-wide based technology, where if you have probes that target each of the neighborhoods in the genome, you can detect if there's actually less number of genes or more numbers of genes for that region than there should be. And essentially it's part of a ratio that when you notice that there's a deletion, and you can notice it in this case, where the signal drops to the left, that essentially means that there's a missing piece of genetic material there. And the technology has developed to the point that now we can use single nucleotide polymorphisms or sequences that are very common in the population to determine how related the parents are, even when you're doing the test. So not only does it give you information about missing neighborhoods, but it tells you how related the family has been, which may increase the risk of recessive diseases, because if you have 2 regions of the genome that are identical in sequence that came from both parents, there may be a higher risk or a chance that if there is a mutation within that region, that both copies, right, both of moms and dads are going to be identical, therefore that there will be a recessive disease that's manifested. So it's a very useful technology to assess both copy number changes and relatedness in the family. So how does this work? Essentially, copy number variations occur because during gametogenesis, when you're making sperm or eggs, there is a piece of those, and I'm using blue here to address or to classify the male or the father. Each of you can sort of self-identify with the colors as you want. But essentially, we'll blame the dad for this one. So in the dad sperm, you usually see a missing piece that occurs as the sperm forms, and then when that gets passed down to the next generation, you have a child that has a missing neighborhood and therefore predisposition for autism. We had described, and I'm just going to give you one example of hundreds of these conditions that have been described in the last quarter century, but this is one that we described many years ago. And what was interesting was that these were 2 children that presented initially or received the diagnosis of autism at a very young age. But then by the time they started entering puberty or later on, they developed psychosis. And to us, it was interesting that it was during puberty that all of a sudden this behavior started to appear. And when we looked carefully at their genetics, what they shared was a missing neighborhood in chromosome 3 in region Q29, so 3Q29. And you can see that the signal gets dropped in both patients. And it turns out that within that region, there are genes that we know are important for establishing the connections of neurons and for maintaining those proper connections. And we knew that 2 of those genes have already been previously suggested from other studies, genome-wide association and otherwise, as potentially causing both the psychosis and the ASD predisposition. And we were very encouraged when large studies that looked at just schizophrenia by itself confirmed our finding, because they actually found independently that the most common copy number change in those individuals was the absence of this region. So what this also started was this conversation in the field that genes that cause autism may actually be involved in many other behavioral problems, not just neurodevelopmental concerns, right? So behaviors like bipolar, schizophrenia, and other ADHD, all of those may actually be part of a large continuum that depends on the same genetic networks and gene etiologies. So very pioneering studies, and this is one that Dan Geschwin published many years ago, where he actually demonstrated for the first time that there was a strong association of copy number changes to autism. And in fact, what he found has been confirmed over the years, that there are a number of copy number variations that are very common, that happen over and over again. And one of them is this 15q duplication. That's one of the most common ones that we observe. This test has some limitations. It aims at identifying these copy number changes, but it will not see changes that are present in low levels in blood, which sometimes means that the missing neighborhood is not in every cell of your body. It's in some of them, but not others. That's called mosaicism. It will not detect sequence amplifications like Fragile X syndrome, which is due to an expansion of a sequence that is trinucleotide repeat. And it will not detect things where there's no net gain or loss of genetic material. So if two chromosomes have swapped and there's a break of a gene where the two chromosomes came together, right? If there's no net gain or loss of material, you will not detect it, but there will still be potentially a role for that gene in causing the patient's symptoms. And of course, it doesn't detect point mutations because it's not sequencing. It's just telling you, is this neighborhood there or not? So this takes us to one of the more other common single gene associated causes of autism. And Fragile X is one that is quoted very commonly because up to 1% of males that present with autism may have a Fragile X gene alteration. So we know that it's on the X chromosome, it's the FMO1 gene. It's a sequence of CGG repeats that gets amplified. And when it gets amplified, it actually causes methylation changes that then lead to expression changes in the brain. And we know that this is important to test for because there are implications for the parents who may carry the premutation allele as well. So we always test as a first tier, as a first pass, particularly males, for the presence of this expansion in the Fragile X gene. But when we talk about single genes and their association with autism, what has emerged over the years is that autism ends up being associated with physical findings that can sometimes be very telling of a specific genetic change. So one that we have an interest in both clinically and in research is this association that was described many decades ago, that a subset of patients with autism will have accelerated brain growth. And for a long time, a subset of them presented with head circumferences that were 4 or 5 standard deviations above the mean. So we're talking about extreme macrocephaly. And the question was, what is causing this? Many times these patients go through hydrocephalus evaluations and they're normal. They have wide ventricular spaces, which are normal, but their brain continues to grow very fast. And it turns out that if you test these individuals for mutations in a gene called P10, you can find a high yield of testing results back. So P10 is actually the most common cause of autism associated with a child that has extreme macrocephaly large head. So we always test. Any child that you see in your practice that has an accelerated brain growth should get this testing included. But why is it important to detect it? Because it turns out that a lot of these patients will have cancer predisposition. And what they look like is just a child that has ASD, and this is one that we evaluated recently, where the head circumference is very large, and essentially they don't have any other symptoms. But what's brewing is that they're developing thyroid nodules that can turn into cancer as they're children. And they start developing polyps in their teenage years that by the time that they're in their 20s need to be screened for, right, in the colon. So this pathway has become, or the gene P10 is part of a pathway called the PI3K-AKT-mTOR pathway, and you'll notice that it's an inhibitor of the pathway. It's the brake that allows this growth-promoting pathway to not work. So when you have a mutation, what happens is this pathway works too much, and by working too much, you have accelerated brain growth. But it also is associated with accelerated growth of other tissues, which then may potentially lead to cancer. So there are some subtypes of autism that need early detection because they would benefit from this early surveillance. And we have a clinic where we actually follow these patients and implement these surveillance plans. And most of these kids end up getting their thyroid taken out because they do develop these thyroid nodules and lesions, the ones that present with autism, over the first years of life. So PTEN and cancer predisposition, we know that mutations in PTEN later on can lead to other manifestations, and it's not clear to date whether the patients that have autism will develop those later on, but we know that, for example, women that have these mutations in later life have a higher incidence of breast cancer, among other symptoms. And the good thing about this is that there's already some sporadic cases where treatments that were used for treating cancers that have mutations in this gene, when you use the same drug to treat the patients that are born with the mutation everywhere in their body and present with autism symptoms or seizures, that treatment with this drug, which is called sirolimus, actually makes a difference. So this is encouraging because it tells us that if we detect these genetic changes early enough, we may potentially allow these patients to have access to therapies that are coming down the road that may actually have an impact not only in the cancer predisposition, but also in their neurodevelopmental outcomes. And the studies are ongoing. So we were very interested in looking back at what is the yield of genetic testing when you look at kids that have a big head, particularly if they have autism. And it turns out when we looked at seizures, language impairment, motor delays, hypotonia, none of that made a difference. What made a difference was their brain growing too fast or was their brain growing too slow? So children that have macrocephaly or microcephaly, a small head, should be a red flag because they most likely will have a predisposition that is genetic and therefore would benefit from testing. So far I've given you examples of single genes where the physical findings are very telling and can actually direct your testing, or using genome-wide microarray tools to find copy number variations. It wasn't until the advent of massive parallel sequencing that we were able to then find the genetic causes for the remainder, which was very challenging because these cases were not familial, were sporadic, and the genetics was very difficult to do. So there were two studies that came out in 2009 that essentially broke the wall of how do we use this technology to diagnose rare diseases. And they were proof of principle that you could use what eventually became exome sequencing in clinical scenarios to diagnose rare diseases. So what is the intent of this technology? The idea is that how do you find mutations that are present in the child where there is no family history of autism or any other developmental disorder? Most of the time, what the literature has identified is that this is due to de novo mutations in the family. So what is a de novo mutation? Again, we're going back to gametogenesis. And if you're making sperm, we'll blame the dad again. So in the dad's sperm, there's actually a mutation that happens, and that mutation gets passed down to the next generation, but neither parent contributed that mutation. It actually happened during gametogenesis. So clinical exome sequencing is the now clinically available diagnostic test that emerged from that technology that allows you to sequence essentially the protein coding portions of the genetic material. That part of the DNA that makes proteins, and therefore that may be most important for body function. So 95% of the gene coding sequences is included. It includes the mitochondrial DNA, which is separate from your nuclear genome. And it's very expensive, depending on where you go. It takes a long time to finalize the answer. And it requires consent prior to sending because there are some implications when you include parents and child for that genetic information. So genetic counseling is a very important part. And what we're trying to avoid is, for example, when you do a test where it includes the parents and the child, you will find out non-paternity, right? So if the dad is not the dad, we're going to end up telling them. And I've had more Popovich moments that I really don't want to live again. So we always consent the family to let them know, especially in private with the mom, we're about to do this test, so make sure that you want to proceed with it because it has that implication. Consanguinity is another one, and whether it's consanguinity that's known or consanguinity that is illegal will actually be identified. And of course, incidental findings. We're looking for changes in genes that may explain the neurodevelopmental concerns of the child, but we may stumble upon a change in a gene that causes heart disease or something that the family wasn't aware of, and now they may be surprised that they have another genetic disease that they were not expecting. So this obviously provides reproductive options and addresses psychosocial issues associated with testing. So the typical story where this is actually important is that exome sequencing has become the way to reach a diagnosis when you have had years without a diagnosis and unexpected clinical findings that no one can really pinpoint to the cause. And it's ideal for situations like autism where you have so many genes that can give you the same symptoms, where identifying the one that is actually affected may have implications for care. And of course, atypical presentations where the gene doesn't follow the textbook. So a lot of studies have been done where they've actually identified the high yield of de novo genetic sequences or changes in causing autism. And in fact, that has actually extended to intellectual disability, ADHD, and other neurobehavioral disorders. So what is the approach? So just to summarize, essentially when you do initial testing, we pursue chromosomal microarray and Fragile X testing. If you have an answer, you stop. If it's negative, 25% of the time you'll have an answer. If it's negative, what you do is you move on to clinical exome sequencing. And clinical exome sequencing may give you a diagnosis or it may not. For that, what we recommend is what we call reanalysis, where the sequence can be looked at again over time to see if there's something new that we can identify. But what is it that we need to know about the genetics of autism? So there are several gaps. One is, what is the diagnostic utility of genetic testing? What is the clinical utility? How does it actually really alter or manage care? And is the genetic testing approach cost effective? Is what we're doing making sense in terms of costs? And the reason that we want to address that is because if you start adding up all the costs between Fragile X microarray, gene testing, and whole exome sequencing, it becomes expensive very quickly. So you want to make sure that you're doing something that has impact. We know that some data is emerging where the impact of this testing is really being validated, both financially and in terms of diagnosis. And in some cases, up to 72% of the time, the diagnosis made a significant difference. So what happens when genetic testing does not identify a clinically significant variant? What do we do next? So I'll give you an example very quickly of a family where at 2 years of age, the individual had presented with autism symptoms, and the exome was negative. But then by 4 years of age, the patient had progressive regression, developmental delay, and brain MRI changes. And then it wasn't until after the reanalysis 2 years later that it was the new symptoms that allowed us to find the genetic cause. And it's actually a disease that is progressively deteriorating. So this is an example where autism can be an early symptom of something that later on can have significant clinical consequences. So reanalysis is essential for understanding the final etiology. So just in the interest of time, where are we going next? The hope is that new technologies like whole genome sequencing, RNA-seq, and metabolomics will actually add to identify new potential etiologies to that 44% where we still don't have an answer. So the recommendation that we're proposing is that clinical exome sequencing be really an essential part of the evaluation of all these children because of the broad genome approach to identify them. And there are many different groups that are pursuing genome sequencing, not just exome, but the whole genome. Those dark areas that we thought were junk, now people are actually looking at them to see, is there something interesting there? And the idea is that now more and more coupled with RNA sequencing, we're finding genetic changes that are hiding in that dark matter that normally we couldn't execute before. So in the future, we'll need to update natural histories of these conditions, see what the role of genome sequencing adds to the diagnosis, provide multidisciplinary care to families when they have a diagnosis of what to do next, and design clinical trials that are good for identifying therapies. And I just want to close with the clinic that we have established at UCLA that aims to do that. It aims to bring experts in psychiatry, in genetics, in social work, psychology, and neurology to address patients that have rare genetic etiologies associated with autism. Thank you.